Dear students, myself Dr. Fatima Sultana, Associate Professor in Department of Zoology, Janki Devi Government Girls College, Kota, Rajasthan. I would like you all students to be safe at home, stay at home and be safe during this pandemic circumstances of COVID-19. We will be providing you online classes and today's topic is mitosis. Mitosis is the process in which the nucleus divides to form two nuclei. Development of an organism occurs by mitosis. Every organism starts its life from a single cell, that is zygote. Repeated mitosis in zygote leads to the formation of the whole body. The process of cell division, which results in the production of two daughter cells from a single parent cell. The daughter cells are identical to one another and to the original parent cell. Now, what are the three reasons why the cells reproduce? And it is a simple process of asexual reproduction in which one cell divides into two cells which are identical to the mother cell. There are three reasons: growth, repair, replacement. The growth of multicellular organisms is due to mitosis. Every multicellular organism multiplies by the process of this mitotic cell division. Healing and regeneration. This also takes place due to this mitotic division. Mitosis produces new cells for healing the wounds and for regeneration. Reproduction also takes place. Mitosis brings about reproduction in unicellular organism. It is just a simple multiplication from which one cell multiplies into 2 and 2 to 4, 4 to 8. It goes on so that a multicellular organism develops. Now, have you ever imagined that how do little elephants grow up to be big elephants? When, as I have told you, that from a single cell, that is zygote, every time the organism, the cells will be multiplying to form a multicellular organism. So slowly we see that the zygote grows, and after a few months and years after, we see that the little elephant changes to the big elephant. Why do animals shed their skin? There are certain animals, for example, like reptiles. They grow in size every year, every time, whenever they grow, the skin doesn't grow because the skin is made up of keratin. So we see that they regularly shed the skin and a new skin is developed on their body. So, because of this regular growth, the body is growing due to which they have to shed their skin. The term mitosis was proposed by Fleming. Mitosis is divided into two main stages: karyokinesis and cytokinesis. Karyokinesis is the multiplication of the nucleus, the division of the nucleus, and cytokinesis is the division of the cytoplasm present in the cell. Mitosis produced genetically identical cells which are similar to mother cell. Karyokinesis is a biological process involving the division of a cell's nucleus. Division of nucleus in mitosis is indirect. It is divided into four stages: prophase, metaphase, anaphase, and telophase. And then comes the last process, cytokinesis, in which the cytoplasm divides. The first, which I have mentioned in this interphase, this is actually the preparation of the cell for the mitotic division, in which there are different phases in the where the DNA synthesis and multiplication of the various cell organelles, the number of the size increases. Interphase, the cell grows in size and prepares itself for division. The DNA gets replicated, organelles replicated, cell increases in size, and metabolism of cell is increased during this phase because there is a continuous synthesis of the proteins and the nucleus which is required, the chromatin material which is required for the further mitotic cell division. We can see the animal cell and plant cell. There is a difference between animal cell and plant cell, that a cell wall is present in the plant cell, which is made up of cellulose, whereas it is absent in animal cell. Prophase, this is the longest stage and the first stage of the mitotic division, which follows the S and G2 phases of interphase. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. Chromatin threads condenses to form chromosomes, and the centriole begins to move towards the opposite poles of the cell. Astral rays forms when the centrioles move to the opposite direction of the cell, or that is opposite poles of the cell. We can see in this diagram also that the pair of centrioles, they are moving opposite to the poles and astral rays are formed. In higher plants this is absent. Astral rays are absent and this is known as anastral mitosis, whereas in animals astral rays are formed and the mitosis is known as astral mitosis. As I have told you that this is the first step of mitosis. Mitosis begins and the cell begins to divide. Centrioles appear and begin to move to opposite ends of the cell. Spindle fibers form between the poles, to which further the chromosomes will get attached. You can see in this animal cell and plant cell clearly the condensation of this chromosome material is seen. They are getting condensed to form chromosomes. Metaphase, the cell prepares chromosome for division. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis, which is known as metaphase. Chromosomes line up at the center of the cell. Spindle fibers attach from daughter cells to chromosomes at the centromere. We can see in this diagram that every chromosome is having two sister chromatids, and they are attached by a centromere. The spindle fibers get attached to the kinetochore of this centromere, and one sister chromatid and the other sister chromatid will face opposite to each other so that they can be pulled towards the opposite direction at the end of the poles. The metaphase is characterized by all the chromosomes coming to lie at the equator, with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. The plane of alignment of the chromosomes at metaphase is referred to as the metaphase plate. We can see clearly that all the chromosomes are aligned in the at the equator of the cell. This is the stage in which we can clearly study the chromosome structure, where the chromosomes are clearly visible. Chromatids attached to the spindle fiber. In this direction diagram you can clearly see that the centrioles are on the opposite direction, that is at the end of the poles opposite to each other, and the centromeres are at chromosomes of the centromeres are attached to the spindle fiber and they are lying on the equator. Animal cell and plant cell, we can see that the chromosomes are aligned at the equator in one line. Clearly the chromosomes are visible. Anaphase, the chromosomes divide. Now here we can see that the spindle fibers pull chromosomes apart. One sister chromatid is pulled to the one end and the other sister chromatid is pulled towards the other end. Centromeres split and chromatids separate. So half of each chromosome moves to each daughter cell. Chromatids separate and begin to move to opposite ends of the cell. In this diagram we can see that the sister chromatids are pulled towards the opposite end of the poles in both animal cell as well as plant cell. Telophase. In this, we can see that the chromosomes have moved apart, the sister chromatids have moved apart, and the nuclear membrane starts developing. Actually, we can say that this is the reverse of prophase. In prophase, the nuclear membrane and the nucleolus and the organelles were disintegrated, whereas in telophase, they start appearing. The nuclear membrane forms around the chromatin material, and two nuclei form at the two ends. And the cell wall, in case of animal cell, it pinches from the center. We can see that there is a cleavage furrow in which the cell is pinched so that two ball-like structures are seen, in which one will form the one daughter cell and the second one will be form the second daughter cell, and these two daughter cells are very identical to the parent mother cell. In plant cell, there is a slight difference because there's a cell wall is present which is made up of cellulose, so the cleavage furrow or the pinching of the cell wall is not possible. A plate is formed in between the two nuclear material which has been moved opposite directions, and the plate it starts developing from the center towards the outer region. So in case of animal cell, we say that it is centripetal mitosis, mitotic division, whereas in case of plant cell, we'll say it is of a centrifugal order. The cytokinesis occurs in centrifugal order, whereas in case of animal cell, it is in the form of centripetal order. The two nuclei are formed in the telophase. It is clearly seen. Chromosomes appear as chromatin threads rather than rods, and mitosis ends up at this stage of telophase. Cytokinesis occurs after mitosis. Cell membrane moves inward to create two daughter cells, and each with its own nucleus with identical chromosomes. There is a difference between mitosis in plants and animals. Plants, in case of plants, centrioles are absent, whereas in animals, centrioles are present. Esters are not formed in case of plants, whereas esters are formed in case of animals. Cell division involves formation of a cell plate. In animals, cell division involves furrowing and cleavage of cytoplasm. Mitosis occurs mainly at meristems, where there is fast growth, the tips of the shoot and root, we can say. And in animals, it occurs in tissues throughout the body. We can take a short recap. Interphase is the stage where the cell prepares itself for the mitotic division. And first stage is the prophase, second metaphase, third anaphase, and fourth telophase. In these four stages, the nuclei divide from one nuclei into two nucleus, and finally in cytokinesis, the cytoplasm divides so that equal portions are given to the two daughter cells. We can see clearly here. This is for your revision. I have mentioned all the things in the boxes, that how the process of this mitotic cell division takes place. Thank you so much.